Thanks so much for being here. We have a bunch of people in the room and a whole bunch of people on WebEx. So we're so excited to have you here. And from here, I will turn things over to Dr. Mark Wainwright, who will introduce our speaker and our lectureship today. Thanks so much.
Good morning. Welcome to the 29th Annual Herman and Faye Sarkowsky Lecture in Child Neurology. Visiting professor today, we're honored to have Dr. Annapurna Poduri from Harvard and Boston Children's Hospital. I want to say a few words first about the Sarkowsky family who made this lecture possible. Herman and Faye Sarkowsky were married for 62 years. Mr. Sarkowsky died in the early 2000s, but they have a long history of philanthropic activity in Seattle to various arts organizations, but also to Seattle Children's and the Division of Child Neurology in particular. Herman Sarkowsky was a part owner and co-founder of the Seattle Seahawks and the Portland Trailblazers. Mrs. Sarkowsky and Herman Sarkowsky made possible an endowed chair, which I hold, a fund which supports some of the academic activities of the Division of Child Neurology, and this lectureship, this visiting professorship in particular. The first of these lectures was given in 1995, and the only criteria for its selection, which is made by the Division of Neurology faculty, is someone who's made substantial contributions to the field of child neurology. Some of the luminaries in this field have been Isabel Rapin, Scott Pomeroy, William Mobley, and many others. Dr. Poduri belongs in this history and tradition with significant contributions on her part. She completed a bachelor's degree at Harvard, medical training at the University of Pennsylvania, followed by a residency at Children's Hospital of Philadelphia, then joined the faculty at Harvard. She's now a professor of neurology, the associate chief for academic development in the Department of Neurology, and as a scientist, the director of the Epilepsy Genetics and Neurogenetics program. She's made landmark discoveries in the field of her interest in the genetic neurodevelopmental disorders and the role of somatic and germline mutations in epilepsy and developmental disorders, including a landmark paper in Neuron growing out of some observations she made as a fellow about neurodevelopmental disorders and their relationship to cerebral malformations and the role of the AKT mutation. Dr. Poduri has received numerous honors from the American Academy of Neurology, the American Neurologic Association, including the Denny Brown Award and the Dreyfus Penry Award. And she's published over 160 papers herself, including numerous other papers as members of a consortium in basic neuroscience. I'd also like to comment on the other impact she's made on child neurology, a reflection of her generosity as a mentor to junior trainees as well. Her 93-page CV contains 12 pages about her mentees. And many of the papers that she's published are papers that are her as a mentor with career opportunities for trainees. Another tangible and long-standing contribution to basic neurosciences and child neurology. We're honored to have her here today, and we're looking forward to hearing her talk.
Thank you so much, Dr. Wainwright, and it's really an honor and privilege to be here. I am really touched by the invitation and so grateful to be able to be here in person to enjoy your company and the hospitality of the whole group here. So I really feel very privileged to be here in person and grateful for the opportunity. Okay, so I called the talk Genetics of Epilepsy, Bedside, Dementia, and Back, and as Mark mentioned, I really have taken my inspiration for the research I've been able to do the last couple of decades from the patients I saw, starting from training. And this really has been a fantastic journey for me to take those clinical observations, ask questions, figure out which questions were being asked by others and work with them, but also figure out what questions maybe weren't being asked. And so to the residents and the fellows out there, you may think that all of these wonderful luminaries with whom you train know everything, and they know almost everything, but there may be some questions that they haven't had time or tools to ask, and think about that and embrace those questions, because you really can make a contribution. And that's my story. I had actually intended in every way to see patients and work with my more research-oriented colleagues. And I had an observation early on that, well, some of the research I thought really had to be done just wasn't being done. And it wasn't because it wasn't important, but it was because people have lots of things to do, and nobody had really taken on the questions I thought were important. So I took them to the bench. I don't have any conflicts of interest. I do want to say I have a concentration of interest, which is that I focus on epilepsy genetics. I put it in quotes, and I took this picture when I was lucky enough to have some time to walk around yesterday afternoon in your wonderful and sunny city. And I wanted you to take a look at this and think about what do you see? Do you see the Space Needle, or do you see a Calder? And think about how they're both there, right? So I might be focusing sometimes on the epilepsy genetics, and other people might be focusing on the autism genetics, and other people might be focusing on the grass at the bottom of the picture. They're all there, right? It's one city. So I want you to keep that in mind when I'm going to focus really on the epilepsy, but it's one brain, it's one developing brain, and all of the different features really come together. But my bias is on the epilepsy side, since I am trained as an epileptologist and still practice on Tuesdays in that area. So here is our roadmap. I'm going to go through the microcosm, the clinical and research microcosm of epilepsy genetics. And then I'll talk a little bit about functional modeling of both germline and mosaic disorders in zebrafish. I'm just going to give you a taste of what we do, and I'm happy to talk about that later with anyone. And then really focus at the end on translating discovery to practice, because the research is really interesting. Any of us could do it full time. But I always think back to, why did I get into this 20-year detour in the first place? It was really because we have patients who need the science to move forward. So I'll start with the epilepsy genetics microcosm, go through this fish modeling, and I'll talk a little bit about the practice part at the end. So the big question that I've been trying to address is, why our patients have epilepsy? 1 in 26 is the current estimate of the lifetime prevalence of epilepsy. Much of it starts early in life, a little bit in our latter decades because of acquired causes. But when we think about why our patients have epilepsy in pediatric neurology, it's largely neurodevelopmental causes. And so we have young children who start off having seizures as infants. We have others who might start in middle school or high school. And when you think about these families who come to us with new onset epilepsy, what are they thinking? Well, first, how do we keep these awful things from happening again? And secondly, well, what happened? What happened, and why is this happening? And we can try to answer that on a patient-by-patient level, but we can also try to answer it on a larger level. 1 in 26 is an awful lot of people. Why are we sort of at this edge of hyperexcitability, and why are we not all having seizures all the time? We've got a balance that's going on, to put it very simplistically, and these patients, for various reasons, have sort of tipped over into the wrong side of the balance and are having a risk for seizures and recurrent seizures, which is really our operational definition of epilepsy. The other reason it's really critical and important, and one of the reasons that drove me to pursue this research, is that our treatment is empiric. 1 in 3 of our patients don't respond to the empiric therapies, and many of the ones who do respond have side effects from the medications we prescribe. And that hasn't changed, even though the number of medications available to us is far more than it was when I was in training. The numbers are more or less the same, and there's still quite a lot of side effects, and a lot of people still experiencing recurrent seizures. So we can take clues from our patients, and again, this is a sort of reminder to just look at what's around you and look at the data that the patients are bringing. They bring their history, they bring their whole life story and their comorbidities, as we sometimes call them in epilepsy, but they're associated conditions that will clue us in as to whether there's a whole brain problem versus maybe a very restricted problem. But they also bring now, or we obtain imaging, which can give us a major clue in terms of what's going on. So in this little boy who started having seizures in infancy, you can hopefully see here, a little trouble with my arrow, but this large malformed hemisphere. So that right hemisphere, remember you're looking at the right over there on your left, the right hemisphere is quite massively malformed. The gray-white matter junction is very indistinct. It's blurred, it's thick. And it's large. And you might say, well, the temporal lobe doesn't look so bad, and you're correct, that temporal lobe doesn't look so bad. But apart from that, the rest of the hemisphere actually is quite malformed, and we call that hemimegalencephaly, half of a large brain. And the treatment for that is surgery. We used to have en bloc resection. Now there's disconnection, maybe with some debulking. It works much of the time. If you do the en-bloc resection, you need a shunt because of all of the fluid dynamic shifts. And you might say, well, that actually takes the seizures away, but how barbaric does that seem that we were taking out half of a child's brain? Someone is asking to annotate the content. Shall I just approve that?
Yeah, you can tell them no.
Okay, all right. Sorry, I don't know what to do with that. So here we had a clue, though, and I looked at these sorts of patients, and this patient was operated on when I was a fellow 21 years ago. And I looked at the imaging and said, wow, how is that not a developmental disruption? We know our brains develop with our proliferative stage and then migration and then the organization, and this whole brain is sort of catastrophically malformed. Something must have happened very, very early on, right? And I'll show you some histopathology in a moment that sort of supports that. But what was going through my mind week to week in the operating room is how are we just having these surgeries go on and then barely looking at the pathology, and then we're just sort of going on to the next case a week later without stopping to say, well, we understand a thing or two about genetics now. The cancer biologists are figuring out what goes on in tumors. Why can't we do this with this brain tissue, which is still coming out? So you can have these large lesions. You might have a somewhat smaller lesion in an individual who has a later presentation. There we go. So this young woman, who is now actually an employee at the hospital upstairs from me, but she started in middle school having focal seizures when she was playing basketball. She would have these periods of blanking out and staring and so on, and they were traced to her lesion there in the left temporal-occipital parietal lesion. She couldn't have a full resection because her language area was right in the middle of all of that, so she had a partial awake resection. Talk about dramatic and kind of horrifying, and thankfully she doesn't remember any of what was going on in under the drape when we were there interrogating her. But the outcome has been good by modern standards. She's seizure-free many, many years. She's actually down to one medication. She's doing great, but there were a lot of bad years in between. And how do we know this was due to a developmental disruption? Well, we can look at the image. She also has an identical twin who, up until middle school, they were functioning completely the same. And then when she started having these seizures, she really had a decline in her academic performance. And then after the surgery and seizure freedom, she sort of picked right back up. And then they continued they went to college together, graduated together, and so on. So we had this sort of natural experiment, if you will, with an identical twin, sort of really telling us, wow, this looks like something that must have happened early in development, but just in her brain after the twins split. So what did we see in the microscope? So in the hemimegalencephaly, we saw these huge neurons. Pyramidal cells should not be blobs like that. We saw balloon cells, even with eccentric nuclei. And that reminded me of something else I had seen really that was being described in the 90s when I was a medical student, but then really was being published more actively, and that was the same sort of features in tuberous sclerosis complex. And if you look under the microscope of some of the samples from hemimegalencephaly and you look at some of the cases with tuberous sclerosis complex, you might actually think you might have switched the slides, which is exactly what I thought when I looked at some of these and some of the focal cortical dysplasias. I said to Elizabeth Bondoc, the neuropath fellow, I said, are you sure you didn't switch the cases? And that's what really got us thinking, wow, we really could use these clues from the neuropathology and sort of take a more focused look to look at the mTOR pathway and the TSC pathway to see if maybe we can find an answer for hemimeg that might be resembling an answer to TSC, and the short answer is yes, in fact it is, and that shouldn't be surprising if the pathology speaks to you the way it spoke to me. But then what about focal cortical dysplasias? And indeed, they have some of them, the type 2 focal cortical dysplasias have several of the same features, these big enlarged neurons, weird processes kind of going every which way. You really should have a bunch of dendrites and a central axon, but who can figure out which is the main axon in part C or E there? It's very, very strange, right? Our neurons just shouldn't look that way. So we had these clues early on. So, before I get into what we found, I just want to go over a little bit this process of mosaicism or post-zygotic mutation, what we sometimes sort of colloquially call somatic mutation. And so there, I just want you to look at this image here of the zygote up top, which I may not be able to guide you too well. Oops, sorry. So here's the zygote. We all start off as one fertilized egg, one cell, right? Splits into two. You're supposed to completely, with great fidelity, copy over all of the DNA from that one cell into each of the two daughter cells. And it turns out it happens, for the most part, with good fidelity, and all 20,000 or so of our genes and their coding portions do get copied over relatively well. And then you can have the 4-cell stage and the 8-cell stage and so on. And as you're going through, eventually you form this 3-layer embryo, and you have cells that are just destined to become the ectoderm and the neuroectoderm, so the skin and the brain and so on. So somewhere along the way, if you have a not completely 100% reproduction of your DNA, you can end up with a mutational event that can give you what you are left looking at when you do the genetic analysis, which is a variant. So we're trying to get some clarity in terms of the terminology. I'm going to say mutation sometimes when I shouldn't, but the mutation is really what we think of as this process of an error being introduced. And what we see in our data are the variants. And what you can have is this mutational event occurring in the post-zygotic stage. And depending on where and when it is, and I'll show you some examples, it'll give you a smaller effect or a larger effect. So if that mutation is happening in a cell that's already been sort of fated to become neuroectoderm or already fated to become brain precursors, then you're only going to see it in the brain. So here, for example, if it's late enough and you have these further cell divisions and it's already going to be affecting only the brain, you may end up with hemimegalencephaly with half of the brain cells affected. Now, it may happen even later, and you can have a smaller area. Why are we not readily detecting this? Well, except in those children who are having brain surgery, because that's still the best case scenario in terms of seizure freedom for a lot of children. Who's going to access the brain in your patient who comes to you with epilepsy? Most of us are not suggesting brain biopsies for especially routine epilepsy coming into the office, but even if you've got a lesion, there's a lot of work that goes on between diagnosis of the epilepsy to thinking about getting brain tissue. Usually we used to be sampling DNA from our leukocytes. When we did a DNA test, it was a blood sample. Now we're often doing cheek swab analysis and getting cells there, but not brain. So that's why we've been missing it. And then even when we do have the brain tissue, there are some sensitivity issues. You can only detect down to a certain number of cells. So I'll go through some examples there. So the early discovery that we had, and this was sort of looking at the first handful of cases that we had accrued from our neurosurgeons who had been collecting epilepsy tissue samples as controls for their brain tumors. And I said, oh, that's maybe not the best idea. The cells we're seeing in these samples they're not tumorous, they're not proliferative lesions by any means, but they certainly don't look normal. They look quite perturbed, and we're wondering if the biology now might be shared between the mTOR pathway, which is an oncogenic pathway, growth and regulation in the normal circumstances, but when it is overactivated, it can lead to tumor production. That's maybe not the best controls for the brain tumor studies, but thanks for collecting it, and we were able to get some tissue, and we looked, and again, I mentioned there's, these old surgeries were full disconnection and debulking of the brain, so that's horrifying. I actually, you do it long enough and you're seeing these patients clinically, and we'd see a case or two of this a year, I kind of forgot how horrifying it is. And Chris Walsh, my scientific mentor as an adult neurologist who doesn't see this all the time, is like, Anne, that is so dramatic. You have to show those pictures. And I showed them at a lab meeting, and people were like, could we not look at this, PhDs, could we not look at this over lunch? It was like, okay, it's really not that ghastly to me anymore. But, we do become accustomed and numb to some of these dramatic things. But this is what we're asking, we used to be asking families to go through. So anyways, we took the tissue. We found similar findings in the neuropathology, not surprisingly. We did a couple of tumor markers. The Ki67 is a proliferative marker. You see 1% to 2% of the cells look like they're proliferating, but that's not enough to sort of call it a proliferative disorder. And really putting all of this together, what I did first was just to grind up the tissue and to look for copy number changes, because that's what you could do 20 years ago. You could actually do these very crude SNP analyses, and based on the content of DNA on every chromosome, you could see if you had too much, too little, or just the right amount. And what we found was an amplification on chromosome 1q, the whole arm of chromosome 1q. So the first time this happened, you can sort of see these shades of pink are giving you a copy number, and in these first two columns, hopefully you can see there's just a little bit more pink. And that's not enough pink to sort of tell you, well, there's 3 copies of chromosome 1 or chromosome 1q. The left here, this is the pictogram of chromosome 1. Here's the P arm and the Q arm. And this pink down here is just a shade pinker. And we could do that. We'd actually get some numbers. And it looked like it had a copy number of 2.5. Well, you can't have 2.5 copies of part of a chromosome, right? So some of the cells had 2 copies and some of the cells had 3 copies. We hypothesized that it was an early event that gave us a partial trisomy. It turns out now with single cell analyses and so on, we were not quite right. It's actually a partial tetrasomy event that led to 4 copies and an even smaller number of the cells, but we didn't know that 20 years ago when we were first grinding up these tissues. So that was really interesting. It didn't give us a mechanism, but 2 cases out of my first 8, I thought maybe that was just sort of dumb luck, and how do we validate that? So I went over to the cancer biologist who sort of taught me how to do quantitative PCR, and we really showed, and again and again, this was not an accident. In different parts of those hemispheres, indeed, there was this increased copy. Well, how does an increased copy of chromosome 1q affect you? Well, if it's in all your cells, you really don't survive. You might survive to live birth, but there's all sorts of cardiac complications, other complications. We looked in the blood of some of these individuals. It wasn't in their blood. That wasn't a surprise. But how is that causing brain overgrowth? Well, there's a couple hundred genes there, and so we could, and we did, start to sequence them in the brain tissue of the other cases. Maybe we'll find our answer from a point mutation. Any of the genetics folks in the rooms know that that's a long endeavor and doesn't always lead to anything. And then we also have this complication of well, we can't see small changes if they're only present in a handful of the cells. So those copy number changes informed us about where to look. I said, okay, let's look at the genes on 1q. But I'm just going to tell you why it's so hard to detect these variants. If you look at something that's in all of your cells, let's say somebody has a heterozygous variant here, and what it should be showing is an A, which shows up in red, a little red peak. And the individual has an inherited change from the mom that's showing up as a G or a gold peak. If you do Sanger sequencing in the lab, which every undergraduate now learns how to do, this is what you'll see. But that's something that's present in every cell of the body. So you can sample the cheeks cells, you can sample the brain cells, you can sample the blood cells. Anywhere you sample, you'll find the same thing, and that's what we call a heterozygous variant, and it looks like this in the old Sanger sequencing, and if you look at next generation sequencing data, you get a bunch of reads, you get recurrent reads, and you can see about half of them are A, and half of them are G. Well, that's very nice if you're looking at a germline variant, but what if you're looking at a somatic variant? And that doesn't matter whether it's inherited, as it is on the left, versus de novo, meaning, okay, it's not here in mom, not here in dad, but it's there in the child. The reads look the same. But what if you're then looking at a somatic variant, and it might look like this. So if you've got a change, let's say a point mutation in one of the many genes on chromosome 1q, which is where I was looking, what you end up with is this very little peak here, which if you've got beautiful clean sequencing, and any of you have done this know it's never this beautiful and clean, but if you had beautiful clean sequencing, you might see that if you knew exactly where to look. But if you were looking across a whole gene at hundreds of codons, you're not going to see this. It's going to disappear into the background. And if you're looking at the reads down here on next generation analysis, you might see it depending on where you set your sort of sensitivity cutoff. So people will say, well, why don't we just see this all the time? You can if you set your filters to find something that's not present in half or so of the reads. But if you don't set your filters that way, this just falls into doesn't even get detected. So you really have to know not only what you're looking for, where you're looking for, but also the sensitivity at which you need to be looking. So Alyssa DeGama, who's was actually a graduate student when I was a postdoc, and now is a postdoc with me in my lab, really sort of has been focusing on, so how do we get better and better and more refined? But a lot of it is not in the average patient you might see who's getting a blood sample or a cheek swab. You really have to know that you're looking specifically at the right tissue and how to look at the DNA and analyze the DNA of that tissue. So for those of you who look at the variant allele fractions, which may not be all that many, what we translate this into is how many of the alleles, the G's and the A's, are normal versus abnormal. And so the variant allele fraction for a germline change is 50%. The variant allele fraction for a somatic change, the one I'm showing you there, is a 20% change. So 20% of the alleles actually have that. So 40% of the cells have an abnormal read. That's what you see. It's very, very hard to detect with the old methods that we all learned in the lab with our Sanger sequencing. So how did we do this? How did we figure anything out? Really what we did is we knew where to look. And not only did we know the 1q region from the work that we had done, but we knew that there was a gene AKT3 that has a cousin gene, AKT1, and that gene is associated with Proteus syndrome, which gives you overgrowth of a lot of tissues, rarely the brain, but usually skin and bone and cartilage and so on. And very nice work from the Biesecker group showed that not only AKT1 was the right gene for Proteus syndrome, but AKT1 at exactly this spot where you have a glutamate to lysine and E to K change, the 17th amino acid, that's where you look. And that's at the 49th codon of the gene. So we said, well, we looked at AKT3, the cousin of that gene, and we didn't see anything for the reasons I mentioned on the last slide. But what if we really hone in And we look specifically at this spot. And we don't just look at our Sanger sequencing the way we usually do, but let's sort of amp it up and sort of see. And hopefully you can see there's a little red peak here where it really should be gold. And that's when we looked at the bulk brain tissue. You look at the blood, no, it's all gold, it looks fine. Well, how did we split this out? Well, we did some really old techniques. We took the DNA from the human brain tissue, we put it into bacteria. Bacteria only have a single strand. So we split out the reads, if you will, into single strands, and then we could see, okay, you've got 17% of the clones of these bacteria that we made that have the abnormal copy. And 83% have the normal copy, which told us, okay, 17% of the clones have the abnormal copy. So if you then put that back into what we know is the case in humans, you've got 17% of these bacterial clones with the abnormal copy. Well, how does that translate to what we see in the human brain? Well, imagine each of those abnormal copies pairing with one of the normal copies. So you've got all this pairing going on. So about a third, or 34%, or a third of the brain cells in humans had the abnormal copy. Now, we can do this in much more sophisticated ways today than we did back then. But actually, when you want to confirm something, this old bacterial cloning technique actually is a nice way to do it with zero ambiguity. You know exactly what you're looking at because you're looking at the single reads of what's in that DNA. If we hadn't had that clue from Proteus, we wouldn't have found this change for many, many years. So we were fortunate to sort of be asking the right questions at the right time with the right tools that became available. So hemimegalencephaly, we sort of solved that today. I'll show you we can solve a lot of those cases. We see that once or twice a year. You probably see it once or twice a year. It's not the most common epilepsy, and people will say, well, that's, my husband, who's a physician scientist, will say, is that really a public health emergency? I was like, well, no, it's not. But remember, it may not be a public health emergency, but epilepsy still is. We still have risk of SUDEP, risk of falls, risk of various things. And how does this rare malformed brain study inform us? Well, think about focal cortical dysplasia, which I showed you under the microscope can look very similar. And if you look at the classic MRIs of the focal cortical dysplasias, you see this sort of stem to stern from the ventricular zone. If you imagine back when this brain was forming all the way out to the cortical mantle, something went wrong in development. And what we thought went wrong in development is that one post-zygotic change occurred. And if that post-zygotic change affected one progenitor cell, and then all of the offspring, the daughter cells of that, ended up migrating and then interspersing, as we know these clones do, interspersing with the normal cells that don't bear the variant, then what you end up with is this sort of wedge-shaped lesion, as we call it on the MRI. It's really an ice cream cone-shaped lesion if you think about it in 3D. And that's the nidus of the epilepsy in many, many, many individuals. So there's not a week that goes by that somebody with focal cortical dysplasia is coming to the epilepsy clinic. So now we've got this rare genetic really informing our common phenomena that we see in epilepsy. So expanding that logic, we looked not only at AKT3, which is a gene in this pathway, which is cartooned here, and also TSC1 and 2, but all of these other genes. So I'm sort of showing this diagram, and you can certainly draw on the literature that comes from here as well. Bill Dobbins, Rita Mirza in particular, have had a huge role in sort of outlining a lot of these variants. And so the reason we were looking now in other samples for these is because we got together with Jay Shandur and Bill Dobbins and others and sort of said, let's put together a candidate gene list and let's look at all the brain tissue we have that's unsolved and really sort of dig in. So I'm showing you Alyssa's synopsis, but you could also look at Rita's synopsis and other people's synopses of these pathways. And not surprisingly, we found, when we looked for them, variants in all of these different genes. The other thing you may notice is that I'm showing you a handful of variants, but there's some recurrent variants and there's some recurrent hotspots. And I won't get into this, but if you look at this gene, for example, PIK3CA, which is upstream from AKT, upstream from TSC and mTOR, there's multiple cases now with this E542K. There's multiple cases with the E545K. These are hotspots in these genes. So as opposed to the genetics of that's mediated by loss of function, you can have a gene where any number of different changes can produce a phenotype. In some of these genes, there are these hotspot mutations or driver mutations, driver variants, where if you alter the gene at just that spot, you're increasing the activity of that gene. If you increase the activity of these upstream, you're increasing the activity of mTOR, you're getting overgrowth proliferation. The other side of this pathway with DEPDC5 and PRL and so on, those are modulators of the pathway, and they're basically inhibitors. So if you inhibit your inhibitor, then you have overgrowth as well. And I'll just say one thing that sometimes people say, well, these are driver mutations, don't they cause cancer? They do. So if you look at cancer databases, the cancer biologists really got this right early on. They started cataloging every single variant anybody ever saw in cancer in tumor samples in what's called the COSMIC database. Most of these are in COSMIC. Actually, it gives us some validity that to say, hey, these do cause abnormalities. So I have a question that I'll throw out so nobody asks me, why don't they cause cancer in the developing brain? You've got this developmental niche where you're sort of promoting growth and you're promoting proliferation. So we want to allow some of that to happen, but you don't want it to sort of go into overdrive and cause cancer. The nuances of that we don't really know, but I can tell you that AKT3 E17K variant that I showed you in the brain, that first point mutation we found, if one of my skin cells were to divide and develop that spontaneously, and remember, we've got thousands of divisions going on in our dividing cells all the time, not in our mature brains, but in our dividing cells, I would get melanoma. And so what's going on in the relatively permissive niche of the developing brain, such that we don't get a brain tumor, but we actually do get melanoma if we have that in our skin, we don't exactly know. But I think that's what the issue is. It's the niche and the timing and the permissiveness in development and embryology that lets us have those things happen. So where and when really matters a lot. So when in the course of our continued cell divisions and development, it really, really matters quite a lot. So if you have something from the zygotic stage and it's in all of your cells, you may have the whole brain involved. So here's an example where you have an mTOR variant in a child with polymicrogyria affecting both hemispheres, early variant. Whereas if you have the somatic mutation, as I went through before, same genes can, you can affect the same genes or different genes, but in the same pathway, you can have half of the brain cells involved. And then if you look finally at the somatic mutations that are causing these focal cortical dysplasias, you really have to look at the brain tissue. And if you look in the parentheses here, you can see how many cells are involved in the samples we looked at. If you look at the 100% of the cells are carrying the variant, you can look at any tissue you want and you'll find it in 100% of the cells or in half of the reads, if it's heterozygous, which these are. About a third of the cells with the hemimegalencephaly, if you look at the brain, down to about 5% of the cells if you look at the focal cortical dysplasia. So that's harder and harder to detect, and now we're down to sort of 1 to 2%. So it's getting better. It's getting better because our sequencing is better, our ability to preserve the tissue and get more cells in a sample, but also to be able to do single cell sequencing and really distinguish between the genomes of the individual cells has gotten more sophisticated, and we can do that now in a way that is expanding discovery. So I'm going to summarize now what's been going on in the field, and this is not just work that we've done in Boston, but work that's gone on here, work that's gone on in Joe Gleason's group, and several of many of now generation 4 of our trainees. I'm sort of in the middle of that, but there's now the sort of 2 more generations of scientists sort of working on this. The other person who's been very involved is Erin Heinzen, who's now at UNC. So we went from seeing these phenomena in hemimeg and thinking, gee, this must be genetic, to now if you took anybody and you sampled their brain with hemimeg, or in the course of disconnecting and debulking, you took tissue, about 80% of the time you'd find something, and it'll be in an mTOR pathway gene. So only a couple of labs doing this clinically, and it's quite a convoluted series of steps to get it done clinically, but I really think we should do this. But 80% of the time it's solvable. The focal cortical dysplasias, about half the time they're solvable, and I'm just going to briefly say that they're not all mTORopathies. If they look like that wedge-shaped lesion, they're mTORopathies, but we have found glycosylation defects, we find variants in genes that we know to be involved in germline genetics, but now in these somatic mutations, so we have to be a little careful. You can't just say, hey, let's use an mTOR inhibitor for all of them, the way people have maybe leaned towards, and we're putting in proposals for trials for mTOR inhibitors for hemimegalencephaly. I would say be careful with the focal cortical dysplasias because they're not all mTORopathies. And then what about these individuals with normal MRIs or with temporal lobe epilepsy? With normal MRIs, with focal resection, which we do less and less if it's non-temporal, we also have found this SLC35A2 glycosylation pathway defect. And then in not normal MRIs, but with temporal lobe epilepsy, our adult neurology colleagues, Sarkowsky and others, have found BRAF, KRAS, other variants. So you might be thinking, BRAF, KRAS, that sounds like a cancer mutation, and it is. These are genes that are involved in cancer. And again, in a developmental niche, and also in the temporal lobes where we have a small percentage of our cells still proliferating postnatally, you can develop these variants. And for one reason or another that we really don't understand, they're not always causing tumors. Sometimes they are associated with low-grade seizures, low-grade tumors in individuals who have seizures from the temporal lobe, but often not. So we're still needing to figure that out. That's a very, very quick summary of a lot of work done by a lot of individuals. My key collaborators are listed here. Similar work has been done particularly at UW Seattle and also down at UCSD with Joe Gleason's lab. So I want to make sure to acknowledge that work. And it's really been nice to see our various work sort of coming to confluence with these disorders. So that's great if you have these abnormal MRIs. We've really made a lot of progress in the field. We can now, when I see somebody with a new onset seizure, which occasionally we do because we're trying to make sure everybody gets to see all the kids with new onset seizures periodically, I'll sort of sit down and say, let's scour that MRI. We know it's coming, seizures are coming from the left frontal lobe. MRI was normal. Let's look at that MRI. And I would tell all the trainees, look at the MRI. I'm sure you get that from your faculty here, but a normal MRI from who knows where is not normal until you've looked at it. A normal MRI from my own institution isn't normal until I've looked at it, because as the epileptologist, I know exactly where I want to look. I know exactly which side and which part of the brain I want to look at. And you're allowed to call your radiologist and say, can you look at this image? And they might say, and that's volume averaging, never mind. But, do it anyway, because then you can sit there with the family and say, and show them the MRI and say, this is where the seizures are coming from, and I can't find a thing. Or if there is a subtle finding that the radiologist will find, I will sit through and scroll through the MRI with the family and say, do you see how long it took me to find that? Now can you see why it took us so long to find that? Because I do this all the time, and I couldn't find it. So I think it's good to be honest with the families about the imaging and what it can and can't tell us. But a lot of times, there's just nothing telling us on the MRI what's going on. And so then you have either focal epilepsy, or in the case of this little girl, generalized epilepsy starting at age 3, GTCs, generalized tonic-clonic seizures, drop attacks, myoclonic seizures. And the clue from her didn't come from her imaging. The clue from her came from her older brother, who had exactly the same presentation a few years later. And the parents are not related. They were told, well, this is a fluke, it'll never happen again. And there it did. Dramatic, horrific regression. The brother ended up in status epilepticus, ended up, really sort of losing most skills. And the parents said, this is this nightmare unfolding again, what's going on? And through a series of very convoluted events, ended up coming to us for evaluation of an exome report that had a maternally inherited variant in the gene GRIN2A. I said, this doesn't sound anything like GRIN2A. Mom's fine. That is, that can be catastrophic in other ways, but this is not what we usually see at all. And the time course was off. And, by the way, the normal MRI wasn't normal when I actually got the imaging. The cerebellum was starting to atrophy. Brother's cerebellum had atrophied, and we weren't sure what this was. And there was a little asterisk on the genetic testing report with 2 candidate variants in a gene called CAD that is part of a pathway that ends up providing uridine in all of our cells. Really important to have uridine. It's a building block of all of our cells. And it turned out in that particular condition, which hadn't been published yet when we were seeing this girl a few years ago, but it's in this uridine pathway, sort of made sense. Well, uridine you can give orally, and there are other biochemical disruptions where uridine has been given safely. So we knew it was something we could think about giving. And I walked down the hall to my colleague, Jerry Berry, who's a metabolism expert, who had actually been my attending at CHOP when I was a resident back in 2001. I said, Jerry, this just, I'm tempted to just try this. This kid is just going downhill very quickly. He says, there's a more recent version of the metabolism book than you have, which I had from 2001. There's a new book out. I think there's a footnote. Hang on. He pulls it out because he had the preprint, and there's a footnote of a preprint of an article from Austria where they described 8 children with this condition, and they had given uridine and actually had seen an improvement. And they said, by the way, the blood shows this really bizarre thing called anisopoikilocytosis, where the blood cells are crazily shaped and so on and so forth. So there we had a peripheral biomarker. We did blood samples. In fact, that's what she has. And I'll show you the interim. The more recent is actually even better, but she did a complete 180. She's still on some seizure meds, but she has just had a complete course correction. She hasn't had a GTC since we started the uridine several years ago. I just got her annual blood smear report, and then we've got under control. There was a year where the blood actually didn't look quite normal, so we went up above on our dose of uridine. You can get uridine at the GNC, the vitamin store. We've actually been prescribing it and titrating it up based on her blood smear. We don't want to fall behind with the neurological development, but she went from losing all tone, not being able to walk, losing all of her language, to talking, running, swimming, causing a lot of trouble actually behaviorally. She's quite a challenge these days, but she reads. I mean, this was sort of a complete course correction. So it's not every case where we get this sort of a change, but you've got to take the history. You've got to ask what's going on, especially when patients are coming from all over the place and maybe brother is not there. Push the history, ask what else has gone on in the family. And we've had patients similarly with recessive conditions where the first child in the family died in another country, and so there's no records anywhere in our health system at all. So depending on the culture, people may or may not offer that sort of information. So I really sort of say that the clues can come from the MRIs and the brain tissue, but the clues might just come from the history, which is what was the case here. So how do we take all of that and how do we sort of move that towards looking broadly at large groups of patients who don't necessarily have our brain tissue samples and who don't necessarily come with a focal epilepsy where you might even think about that? We started as a community to try to put cases together with what we call developmental and epileptic encephalopathy, not just seizures, but early onset seizures and developmental consequences that we surmised were coming from a common change affecting the brain cells in the growing individual, and could be coming from a number of different genes. We knew a handful of genes in the 90s, like 3, right, really a handful. We knew by 2011 of a dozen or so genes that could be associated with epilepsy, either because we had pedigrees where there were multiple generations of mild enough epilepsy that you could live to reproduce, but also severe cases that were happening sporadically. And we saw them all the time in the clinic, and it was sort of called idiopathic. Well, it must be one of those things. Well, the one of those things was actually genetic one of those things in many of these cases. But it took putting them together as a collective. So we put many, many different cases, over 300 cases together across 26 different sites, and we're able to show statistical evidence, not just, I see this gene, it looks interesting, it's expressed in the brain, maybe this is it, but statistical evidence with, what got a lot of attention as being the first demonstration that these genes were involved in epilepsy, because we did a case control study. For the first time, there were normal individuals without early onset disease who had their DNA collected, and we can compare their DNA to what we saw in this population of people with epilepsy. And if you look at this list, some of these genes maybe you've never heard of, but some of them, especially the one with the biggest signal, is SCN1A. And you might look and say, 26 sites, it's $25 million. Did you really need to do all that to tell us that SCN1A was an epilepsy gene? We knew that 20 years ago. We knew that long before this study was done. We knew that from family studies. We knew that from a lot of data, really compelling data, suggesting that that gene was important. And I don't see that as a waste of money. I see that as a positive control. Fantastic. The gene that we knew was involved in early infantile epilepsy did show up on this. If it hadn't, my goodness, what a catastrophe that would have been. But a lot of these other genes we'd never heard of. I had never heard of PIK3AP1. It certainly hadn't heard of, some of these other ones at least sound like genes that should be involved in ion channel disorders. This gene SLC35A2, if you're a genetics person might have been paying attention, that's that glycosylation gene I showed you with the brain tissue. What's it doing here on this germline thing? Well, the reason we found it in the brain tissue is because it showed up on this study. And Aaron Heinzen, who was a postdoc when I was a postdoc as part of this study, was doing brain tissue work to sort of see if any of these genes had relevance in some of the brain tissue work we were doing. That's sort of the sort of 50,000-foot view. The real story is that we were looking for mTOR pathway genetics in our brain tissue. And we said, well, why not look for these genes? And her postdoc put them all on the same panel, and that's how we found out that some of the non-mTOR genetics was happening. So it all sort of blends together. But this was an early study that involved a lot of people, and it put together a lot of networks of people who are still working together. How do we, look at some of the these are the severe cases. How do we look? I'm going to gloss over this quickly for the sake of time, but what about the non-acquired epilepsies in people who are relatively healthy, going to high school and college and working out there and are, not doing so badly? Well, for that, we looked at initially sibling pairs to say, well, okay, here's two siblings in a family. But as pediatricians, we said, well, we've got all these kids with parents with epilepsy. Don't you want them? And initially they said, no, it's a sibling pair design. I said, well, we've got all these kids whose parents have epilepsy, and those parents survived to have children. They were well enough neurologically to go on to partner and mate and have children. So don't you want to look at them? Maybe that'll tell us about the broader group of patients with epilepsy. So we sort of needled our way in with our parent-child pairs. And in fact, the genes that we found that are the most important for the sibling and the parent-child pairs are DEPTIC5, that gene that's upstream of mTOR, but now with milder variants causing a milder phenotype, and PCDH19, which we see in all these different settings as well, and SCN1A. No surprise, it's everywhere. It's a big gene, so it gets hit spontaneously a lot when you have spontaneous variants. Sometimes they're really bad, and you have a child with no history. Sometimes it's pretty mild and you can have multiple generations. So it's all sort of coming together as a confluence. All of the same genes can be involved, again, depending on when and where you affect those genes in the brain, and also where in the gene you affect the protein. So how do we model some of these? My lab has been using zebrafish as a model. It can be very convenient. You've got a lot of different, a lot of fish, and it'll do a lot of different genetics quickly, and you can look at germline models. So we take a heterozygous fish and take another heterozygous fish with a change in, let's say, DEPTIC5 or PCDH19, and we can cross them, and you have heterozygous, you get a nice couple hundred fish. A quarter of them or so will be homozygous, a quarter of them or so will be wild type, and then half of them will be heterozygous. Good old genetics. That's fine, but we can also, because the way we introduce these mutations now with CRISPR gene editing is just to inject these guides, like the GPS of our gene editing, and then we inject something that will cut, the CRISPR-Cas9 system will cut the DNA. We can inject that in the one cell stage and then create an embryo that has but we can also just wait till the cells divide and inject it into the 2-cell stage and create a post-zygotic mutation, which is exactly what we think is going on in some of the human disorders. So I thought it was a really good way to model some of these genes, and I started with PCDH19 and DEPTIC5 because we were seeing them in our human populations. We were also seeing them in clinic because they were included on clinical panels before we had exomes. So they're really easy to manipulate genetically. They actually do have seizure-like phenomena. They swim really, really fast, like tonic activity, and then they kind of flop over like they're post-ictal. It's sort of sad, but these are little embryos. You can actually not do a full EEG, although we did figure out a way to put a lot of electrodes on, but you can easily put an electrode into their brains and get electrophysiology data. And then you can look at their neurons, and we can even do some measurement of calcium-mediated activity. So this is one of our PCDH19 models where we can look at these neurons, and when there's a big event, an electrical event, you can also see the calcium being expressed as calcium fluorophores, an indication of activity-dependent change. And so that's sort of an illustration from one of the things, but I'll just show you some of the other things that we found. If you look in the middle of these pictures here, right here is where the interneurons are in this larval zebrafish. And what we started to do was to count the interneurons in our wild type, and we started counting the interneurons in our mutants. And on the face of it, it may not immediately look like there's that many fewer neurons. Maybe it's a little shade less. But we counted very, very religiously and counted several animals. And it turns out there's a dropout in the number of interneurons in this model. Why did we think to do that? Well, simplistically, excitation, inhibition, you either maybe have too much excitation, too little inhibition. It sort of made sense. But really why we did that is our friend and collaborator from Yale, Ellen Hoffman, had seen a dropout of interneurons, sort of to the same degree in one of her models. And she was looking at something completely different, but in a brain-expressed gene. We sort of said, well, that's sort of interesting. Why don't we follow that model and take a look? So we've seen this phenomenon now in this model, but also in others, and we're sort of trying to figure out where are the other interneurons. They're not dying. We've actually looked for signs of cell death, and we can't see that there's evidence of cell death. So my leading hypothesis is that they're not developing properly, and that the progenitors are leading to there's some split off of cells that are ending up somewhere else, and they're not carrying the markers, because I've been talking about them blithely as interneurons. And what are they really? These are progenitor cells that express DLX5 and DLX6, which is what interneurons are supposed to express in development. But if something's going haywire early in the progenitor pool, maybe they're expressing something else. So that's one of the unanswered questions. So briefly, we'll take candidate genes that we prioritize from large numbers of papers and human sequencing studies. We'll look in high throughput fashion, so you can stick these little 5-day larvae into 96 well plates, and each of them will swim, and you can look at the swimming activity in them. We can confirm with electrophysiology or with calcium imaging, but the gold standard really is electrophysiology. So I'll just show you what that looks like. Only one trace, so you don't get your nice 10/20 montage that you will with an EEG. We tried with little electrode arrays, which you can do, but is even more cumbersome than actually putting an electrode in the head. But you can see there's different amounts of spikings, different amount of what we call bursting. And we sort of said, well, surely Scott Barabin, who started doing this years ago in the fish, there's got to be a repertoire of abnormalities. And there sort of is and there sort of isn't. So we've actually had to put a lot of computational power into deciding, well, how many spikes in a row is a burst? How many spikes in a row is a run? I mean, we do this in neonatal critical care. It's sort of still dealing with what's a burst, what's a run, what's a seizure. We're sort of redefining it. And my lab folks were a little bit rankled, like, well, how do we not know? I said, well, keep in mind that when I tell you that that's a burst on a human EEG, I've looked at how many thousands and thousands of bursts. we haven't looked at thousands and thousands of these yet. And so we're sort of trying to figure that out. But hopefully I can sort of convince you that sometimes there's a little bit of low-level activity, sometimes there are these higher amplitude bits of activity, and sometimes they kind of cluster. And we've had to define exactly how many in what period of time we're calling a burst, and then look at a lot of animals. But it is nice to be able to have that. So what we've done is we started, and this was work done by Jeremy Allman, who was a postdoc in my lab years ago, who really wanted to do all of these genes. And we said, well, let's look at a smaller list. Let's look at the ones where, if we put some in silico constraints, we've got all this normal human data now available. Let's look at genes that actually don't tolerate variability in humans. So how do we know what those are? Well, there's a database called the NOMAD database, genome aggregation. It started off with 1,000 individuals when I was a postdoc, from 1,000 genomes, but now there's over 100,000 individuals. And so you've got all these data from people who don't have early onset disease. Normal is a word that I try not to use because who knows, But they don't have early onset bad epilepsy for sure. And so we can constrain our list down, and that actually does a lot. That got rid of couple thousand genes, almost more than a couple thousand genes. And then, well, which ones can we really model in the zebrafish? Well, it turns out that they're vertebrates. They have, on average, 75 or 80% homology to us, which is pretty great. But we don't want to just look at the average. We wanted to look at genes where we knew we could try to model these genes in the zebrafish because they had enough homology to us, which our neuromuscular colleagues who've been doing this longer than we have sort of set 60% at a somewhat arbitrary cutoff, but we were hoping for even higher. Then we'll look at their behavior, look at how many are having these seizure-like events, and we went from 40-some-odd mutants that we were able to generate and produce multiple generations of these fish to only 5 actually having these seizure-like events. Now, these were candidate genes, most of which have not been associated with human disease yet. We're sort of disappointed that it wasn't more, but at least it was a few. We validated 3 with electrophysiology and the other 2 actually with these interneuron abnormalities. And so we're sort of putting this together. And Mark LaCoursier, who's in the bottom left corner, is my first tech, whom I hired in 2012, who's been working at this project now for several years, is sort of taking this to the finish line. So here are the genes. I don't know if they mean anything to anybody, but if they do, please let me know. We'd love to put things together. One of them sort of snuck on the list. So KCND2 snuck on because I was doing a human sequencing project in the clinic, and somebody with a very interesting epilepsy had that gene. It just barely, if you round up, makes the cutoff for the homology. And we sort of said, let's put that on it. It's been one of our most dramatically abnormal genes. So, it sort of leaves us wondering if we've thrown some of the baby out with the bathwater with some of the other genes we excluded. We probably have. But this at least sets up a system to do high throughput screening at the front end with the behavior, and then sort of take things to sort of more, I wouldn't say mechanistic, but more sort of network assessments of the electrophysiology and the interneuron abnormalities. Now we need to sort of look at the mechanisms for these individual genes where we've seen good evidence. So how do we take all of this and take this to patients? Sort of talked about brain tissue and zebrafish and all these sort of things, which seem like they're farther away from the clinic than they are. But I would argue that they're really informing our translation and giving us evidence to be able to take genes that are maybe interesting, maybe present in a handful of people, really to the finish line of validating them. So my friends and colleagues here who are not here are at the ACMG meeting, the American College of Medical Genetics. And I completely forgot that was happening, but they've all sent their regards and are now talking about very similar topics in Toronto at the ACMG. I'm not going to go over that because that's a whole week's worth of lectures, but there's a way that we take a gene from, hey, this looks like a gene expressed in the brain that might be causing epilepsy or autism or whatever, and we validate it. And they've actually put together, our genetics colleagues have put together very nice criteria so you can actually do this. It turns out it hadn't been done for somatic mutations in brain disorders, so we did that. That was a lot of work I'm happy to tell you about separately. If you're interested, it's really well worth doing, but a lot of work and a lot of time. But now we have validation, and anybody can now do the same thing with a simple algorithm that's online. So we decided to sort of put into practice what we were doing and try to put all of this together. So we had institutional support through this rare disease cohorts initiatives. They paid for the sequencing. We had to share the data. I said, are you sure you're willing to share all your data? I said, if you look at what I've been doing all these years, of course, I would much rather have somebody else from the kidney cohort find something that I missed than never find it. And that's happening, actually, with a lot of bidirectional collaborations now across 50 different cohorts at the hospital. And the data are available if anybody wants to apply to have them there. We're giving any ownership of the data away by having the hospital pay for it, and we were all very happy to do that. So we started with a little over 500 cases, and we said, let's just sequence anybody. This was unexplained epilepsy, any age of onset, under 18, all comers. Some of the patients were almost my age. They kind of came back from the adult hospital and said, hey, there was no sequencing when we were, when it's usually the parents when my child was born, so can you do this now? The siblings want to know, people want to know. 100 or so, we found an answer. We had clinical, we agreed to do all this, but we said, you've got to give us the resources to do a clinical return of results. So we gave them the answers. 100 had novel genes, which we don't really know for sure, so we're doing experiments to put them into patients. And then we said, well, this is great, but 45-year-old, we find out that they have an SCN1A variant. How great for closure, but how not real time that is. So let's now look at the babies. So we partnered with Toronto Sick Kids, Great Ormond Street, and Melbourne Royal Children's Hospital to really do this in an accelerated fashion and with trio genomes. So we've got the child, the parents. We offer them, if they have epilepsy within the last 6 weeks, in hospital, not in hospital, rapid genome sequencing. We said we can't do this on a research basis if it's going to be rapid. We need to do a parallel clinical process. So we did that, and we actually found answers in almost half of them. And this was work done really led by Alyssa, who was that grad student I worked with when I was a postdoc. And so what we did is we put together this series, and I'm not going to go through every gene because we'd be here another several hours, but just suffice to say, first day of life, 11th month of life onset, a little bit more early on in mid the first year, but even the later ones who showed up at 10 months of age, we're finding variants in a lot of these genes, and we're finding the same genes represented at different parts of that first year of life. And in some, we found 43 of the first 100 to have compelling clinically returnable results, and 42 of those 43 had impacts in medical management, prognosis, and recurrence risk counseling, sometimes very favorable. So if you look at the middle here, this PRRT2 person, I'm having trouble pointing at, that's a great prognosis. The seizures will go away, the development will be fine. That's fantastic to be able to deliver that to a family. Some of them were, really, really, really horrible compound heterozygous variants in recessive diseases where the outcome is dismal 100%, and that's horrible, and our NICU colleagues were like, you're hitting us a little too hard, too fast with this. But it actually did allow them and the neuroconsults teams and the families to make decisions about direction of care in a way that otherwise we wouldn't have been able to do. So lots more happening there. Here's the field. Al George put this together so beautifully in this Cambridge Elements series we're putting together. We went from when some of us were in school to this handful of genes to now more than we can keep track of. I see something new in clinic every week. Either I see or one of the fellows sees or somebody in the group sees. So how are we doing this? We put together this little synopsis of this back and forth between the clinic and the lab years ago at Brandy Fuhrman's recommendation from the NINDS. I thought it was kind of going to be boring to put together. This is what we do. It's led to all sorts of people coming to visit us, which we love. So you're welcome to come and visit us if you care to do so. It's been really, really fun. We took that to systematic review, which was really led by Beth Scheidley, my senior genetic counseling partner in the team. And then we took that really to say, if you're younger, if you have intellectual disabilities, high risk of finding something. The genetic counselors didn't want to wait for the neurologist, so they made practice guidelines about sequencing and unexplained epilepsy. The short answer is do it. You'll find something most of the time. And then the AES was able to endorse that. So with that, I'll just say that we've learned a lot. Know the disease. It starts in the clinic. The clinical observations really led us to be able to change our practice. I'm going to flip through because I know we're close to the hour. Collaboration has really been the mainstay of this. This wouldn't have happened without lots of people collaborating, clinicians, scientists, and across institutions. And really keeping our mission in mind that we were doing this for the long arc of how are we practicing medicine, the mentorship, the diversity of people. As I've mentioned, a lot of students, genetic counselors, a lot of people who are not the typical physicians doing this, and then communication. You've got to publish your data, you've got to get it out there in terms of guidelines and so on. So I hope we'll move from the empiric world to, hey, we've solved your case. This is the pathway that's involved in your epilepsy. This is the treatment we have. Maybe we haven't solved it from the regular ways, but we'll use RNA sequencing or something. We'll figure out which colored box you should be in. We'll use those same treatments. And of course, as I mentioned at the end, there's lots of genes, there's lots of pathways. But I think this is where we're heading. So I will just say that we're in this era of gene-based therapies that are emerging. We really need to expand our access of diagnostics to populations who are really not represented in our cohorts. So we're working actively on that. And all of us can prioritize the key elements that are vital to the science, really drive things forward with really good science. We can't have false associations. We really have to get the right stuff out there. I mentioned the mentorship and collaboration and the diversity already. And I'll just end by saying that you all will have ideas, and you'll have ideas that will be just like this. It's a clinical observation that will lead to some scientific pursuit. Find the right people. Many of them will be here. You've got a wonderful group of people. Some of them might be across the country or across the world. Find them and do the work, and it'll make a difference. And the 20 years have gone very, very quickly, and it's been thanks to a lot of folks who've made it possible. So many, many different colleagues and collaborators, many different national and institutional and philanthropic support. It really does take sort of a bake sale approach to funding research that's different. Just wanted to give a quick shout out to my mentors, but also to the large number of mentees who have done the work. I've tried to show their pictures and highlight some of their publications, but lots of bidirectional mentorship and multiple generations of work involved, and mostly the folks who are at home manning the fort so I can be here visiting and enjoying your company. The stars are for the trainees. We're finally back to in-person gatherings, which we're so happy to do. But it's been really, really fun to share this work with you. I look forward to talking to some of you through the day. And if there's parts that piqued your interest or data that would be of interest to you, please do reach out. We're happy to share and work together. Thank you.